Okay, so shall we? I think we should make a start. So it is my pleasure to introduce Professor Itamar Ronen. Itamar is currently at the University of Leiden Medical Center, which is why he is doing this remotely. I believe he will be joining us permanently here in Sussex. It's sometime in the new year, so we'll see him a lot more on campus. Itamar will be the new director of CISC, taking over from Mara. I don't know a huge amount about his work, so I look forward to hearing about that. But I have had a chance to meet him and hang out with him a little bit, and he's a lovely and approachable guy. So it's going to be great to welcome him here to the Sussex community. So over to you, Itamar.
Thank you so much, Chris. Can you guys hear me well? Great, thank you. Thank you for this opportunity. It's the first time that I give a talk in which Brighton and Sussex Medical School is actually appearing as my affiliation, so it's a big cause for celebration. Again, thanks a lot for having me. I understand that I have you as captive audience for the next hour or so. So I'll try to do the best not to bore you too much with scientific details and kind of vague plans, but dig in directly into things that I hope are of common interest to both you and myself. I will just minimize the thumbnail so I have clear vision of what I'm talking about. The opening slide, this is by no means a description of my itinerary from Tel Aviv to Brighton, and the appearance of Banksy's rat with Welcome to Hell has nothing to do with Brighton at all. I hope it will become a little bit clearer when we go down the path of the talk itself. In terms of what I'm planning for you today, very simply, I want to talk about, to start with the scientific part, a little bit talk about what motivates me as an MRI physicist. And what motivates me in MRI physicist is to increase the specificity of MRI methods to different phenomena that we want to observe using MRI. I will start with giving examples from my venture into microstructural investigations and trying to make them more specific. And then also to things that I actually started as a PhD student, but revisited them later on in my career, which is how to make functional investigations more specific. I will finish with talking a little bit about my personal research plans for CISC, which have nothing to do with my or let's say less to do with my personal interest, but in my wish to integrate my research with the research that is existing on campus already. But let's start with the scientific part, or let's say with the data-driven scientific part, and then we'll get on to the data-free part of the talk. And the question is, why do we need more specificity in MRI? And I want to give a couple of examples as to why is that really necessary. And I will start with, I think, one of the most spectacular use of MRI these days, let's say from the late 1980s all the way to these days, which is the use of MRI to investigate tissue microscopic structures, cytomorphology and microstructure. Now, the link between diffusion and structure is something that has been already shown already in the early days of MRI, already in the 1950s. But to put this type of link to work in living tissue was the idea of several luminaries in the field. Now, people, of course, know the two luminaries that I show here, Peter Besser, who is the founding father of diffusion tensor imaging, and Denis Lebihan, maybe arguably the founding father of biological use of diffusion in MRI in general. And soon after the introduction of diffusion-weighted MRI, it was clear that we can use diffusion in MRI as a probe for tissue microstructure. And of course, what came out are those magnificent, beautiful correlates of tissue microstructure. You can see on the left the so-called color-coded fractional anisotropy map, which tells us about alignment of microscopic structures like, for example, white matter axons in a kind of colligative direction that gives us a link between the fact that the axons are anisotropic structure and the anisotropy of diffusion as we measure it by MRI. And this gives rise, of course, to the spaghetti-like tractographic images that you can see on the right. The problem is that when you actually look at the tissue with a microscope, and well, this is, of course, a kind of a cartoon-type microscope, and not use the MRI as a microscope, we see that the picture is much more complicated to be characterized by a few variables. And the question is, where does the signal that we measure and that gives rise to the diffusion-weighted image comes from? First of all, we have to distinguish between, generally speaking, the intracellular and extracellular space. But we also have a multitude of cell types that comprise tissue, be it neural tissue, but also, of course, tissues outside the brain. Each cell is, of course, a multitude of structural subunits, and this is also complicating the relationship between what we measure and what actually happens on the microstructural level. Compounding everything even more is the fact that our probe is not necessarily compartment specific for a long time, because there is also cross-membrane exchange of what we use as the marker for microstructure, which are water molecules. And there is passive exchange and there is active exchange. And this, of course, makes the picture even more complex. So ideally, we would like to have a situation whereby we take this kind of whitewashed picture where everything has the same color and try to color our information in such a way that we know exactly where it comes from, extracellular space, or which type of cells we come from. Similarly, when we talk about functional MRI, but in functional MRI, we can actually distinguish another type of specificity, not just compartmental specificity, but physiological process specificity. And I put here two of the kind of founding fathers of functional MRI, Seiji Ogawa and the late Jack Beliveau, who created a tool that is, of course, now widely used to tell us about the spatial and temporal behavior of brain function. But if we look at this beautiful image that I found just recently on an archive article by James Rowe and his colleagues in Cambridge, what happens between the actual neuronal activity and the moment in which we detect it using functional MRI is a set of filters or a set of proxies that eventually creates a kind of a disconnect between the neuronal activity and what we measure, but not a complete disconnect, because we know that there is a relationship, but this relationship is much more complex and compounded by a multitude of factors that are spatial, temporal, and process-wise difficult to disentangle. So the bold signal, if you want, the blood oxygenation level dependent signal, is a kind of an MR filtered version of the hemodynamic response to neuronal activity. And some of us are busy in separating contributing process and doing modeling of them. For example, Iris, who is now also a faculty in our school, is an expert on measuring cerebral blood flow, which is a type of measurement that isolates one particular physiological variable, making interpretation of changes with respect to function somehow more clear. But one can also try to get closer to the neuronal activity itself and devise methods that are closely related to neuronal activity, bypassing the hemodynamic response. And this is something that I want to also mention in the later part of my talk. And I want to start with the quest for specificity from, let's say, the bulk of my work recently, which is in microstructural investigations. Again, microstructural investigations are these modes or ways for us to use MR in order to say something quantitative, not just qualitative in the sense of microstructural integrity or ways kind of vague terms that I would be extremely glad if they disappear somehow at some point from the lingo of people that do this type of measurements, but to try to really get into the contribution of the various compartments in tissue to changes to make diffusion-weighted measurements true markers for things that actually happen in tissue. And one way to do it is to combine diffusion-weighted measurements of water with heavy modeling. And I gave this example because you may actually recognize most of all, maybe all the names in this particular paper, in which there is a beautiful use of one approach to get compartmental specificity using water-based measurements to say something about the different compartments in white matter and the changes, the specific changes in the organization of these compartments, in this case, in first episode psychosis. The approach that experimentalists like myself and others, together with modeling people, as you can see from the pictures that just appeared near the title above, is different. And this is not just focus on modeling, but to try to simplify the experimental picture by targeting markers, if you want, or microstructural markers that behave similarly to water, in the sense that we can measure them using MR and combine these measurements with diffusion weighting, but reside in particular tissue compartments. And this type of measurements is provided to us by what we call magnetic resonance spectroscopy. In this case, proton magnetic resonance spectroscopy. The magnetic resonance spectroscopic spectrum of the human brain is this trace that you see here. That is a kind of a little forest of different peaks, but those different peaks or this forest can be decomposed into the individual contributions of the various resonances that we know can be attributed to specific metabolites. And those metabolites are in the millimolar range, so we can measure them using MR in a decent signal-to-noise ratio. So we can not only measure them, but we can eventually also apply diffusion weighting to them and measure how do they diffuse within the intracellular space. So we have class of metabolites that we know that are almost exclusively residing in the intracellular space of neurons, whereas other metabolites like inositol or choline compounds, or here the class of neuronal metabolites comprises N-acetylaspartate and the excitatory neurotransmitter glutamate. And in the glial compartment, we can see another group of metabolites. Mostly what we measure is those two main metabolites, which are inositol and total choline compounds that reside mostly in glia. And then we have our blunt tools, which are essentially spectroscopic sequences to detect those spectra, aided with or added with diffusion-weighted modules, similarly to what we do in diffusion-weighted imaging. And then once we get the diffusion-weighted spectra, we can use the data to obtain information about what happens in the intracellular space at different time scales. We can measure at very short time scales and obtain information about, for example, cytoplasmic viscosity or tortuosity within the cytoplasm. But if we go to longer diffusion times, we can measure things that are related to cellular morphology. We can hope to get some information on subcompartmentalization and compartmental exchange. And I will later show you that this is also a tool to get us information about chemical exchange and metabolism. But bear in mind and don't forget that the signal that we get from metabolites is about 3 to 4 orders of magnitude lower than what we get from water. So we have a very limited spatial resolution. So we typically measure our diffusion-weighted spectroscopic measures on big boxes and try to infer on microstructural features from big boxes, which is, of course, a feat in itself. And I want to tell you a little bit about my journey in this type of investigations. I will start with the first, or let's say kind of the derivative of the first study that I did with diffusion-weighted MRS. I started doing diffusion-weighted MRS study when I was in Boston University using a 3 Tesla scanner, but then came an opportunity to come to Leiden and perform measurements on a 7 Tesla scanner. And I naively thought that it would be interesting to just show a proof of concept. And a proof of concept would be to take our big box and go to a white matter tract that is reasonably well coherent and well organized, which is the corpus callosum, and to go to an area of the corpus callosum where the fibers run, roughly in the same direction, meaning connecting the two hemispheres. So in the left-right direction, when we look, for example, in this image here, and to perform two sets of measurements, one that goes perpendicular to the direction of the propagation of the cross-callosal fibers, and one in parallel to it. And these are typical spectra that one can get from it. You see the blue trace is a spectrum of the non-diffusion weighted measurements from this big box on the corpus callosum. This is the N-acetyl aspartate, the axonal constituent, and these are the creatine and choline, which also exist in significant amount also in, for example, astrocytes and oligodendrocytes and microglia. And when we measure the attenuation of the signal as a function of diffusion weighting for those three metabolites for creatine, choline, and then acetyl aspartate, well, you can see that the measurement perpendicular to the fibers gives rise to a relatively shallow decay of the signal. That means that the diffusion perpendicular to the fiber propagation direction is relatively minor, of course, because diffusion is blocked by the axonal membrane. But when we measure parallel to the fibers, the diffusion coefficient, which is essentially the slope of the logarithm of the signal as a function of the diffusion weighting, is much steeper. So this gives us a kind of a feeling of the macroscopic anisotropy of fibers, in this case of axons, in the corpus callosum, just by the difference between those two, the difference in diffusion coefficient as would be calculated from those two curves. And you can see already with your kind of in the naked eye that when we look at the metabolites that are not purely axonal, that exist also in, let's say, isotropic structures like astrocytes and oligodendrocytes, then the difference in the slopes of those two curves is much smaller than what happens in the N-acetyl aspartate. And when we calculate, let's say, the poor man's fractional anisotropy, which is essentially the ratio between the diffusion coefficient calculated from the perpendicular direction and the one parallel to it, and you can see that if the NAA gives us a factor of almost 5, then the creatine and the choline that exist also in outside the axons give a much smaller difference in those diffusion coefficients. So this is just a representation of the macroscopic anisotropy of axonal fibers in the corpus callosum via the strong anisotropy of NAA, diminished by presence in other structures that are isotropic for choline and creatine. So there is a strong differential effect of macroscopic anisotropy across cell types. But here is the catch, is that we can actually use this information to infer something about the distribution of metabolites across cell types. And this is something that we cannot do almost in any other kind of measurement when we do it non-invasively or in vivo in a living system. So if we assume, for example, that the N-acetyl aspartate is 100% axonal, and that axons are the only contributors to this macroscopic fractional anisotropy, meaning that the fractional anisotropy of glia is 0, then we can simply say that the axonal fraction of the metabolite would simply be the ratio between this directional ratio and the directional ratio of NAA. So if we assume that the fractional, the axonal fraction of NAA is 100%, then you can see that for the creatine, we get roughly 50% of the creatine in the corpus callosum in this volume resides within axonal fibers, and with the choline, we get even less than that. So first of all, this is kind of a demonstration of the usefulness of this type of measurements, not for microstructural purposes, but for a purpose that is completely outside the microstructural investigation. but it's also something that gives you already a sense that when we say cell specificity of metabolites, one has to be really careful. The certainty about the specificity of NAA2 axons is quite well established, but you can really see that when we say that choline is a glial component or glial metabolite, this is absolutely not true. So we have quite a significant fraction, if you want, 54% of the choline, at least in this volume, is within axons or within neurons. So one has to be careful about this type of statements. This measurement that I performed on the corpus callosum kind of, promoted an idea that it would be actually interesting to try to model the information on the diffusion of NAA parallel and perpendicular to try to get some information on the orientation dispersion of axons within the corpus callosum from this measurement. This is kind of a tall order to do it from such a kind of big voxel measurement, but I was up to the challenge, and I was encouraged to take this challenge by the guy that appears here in the picture. This is Matt Buddy from Medical College, Wisconsin, who saw a poster of my results in one of the ISMRM conferences and said, I can give you some post-mortem validation for whatever model you want to use. Why don't we work together? And I said, well, that's a good idea. That's an interesting and productive idea. So the idea was to use the diffusion of N-acetyl aspartate alone in perpendicular and in parallel to the axonal fibers to try to estimate two things. One is the diffusion coefficient of N-acetyl aspartate, the so-called cytosolic diffusion coefficient of N-acetyl aspartate. And the other thing would be to try to estimate the dispersion orientation standard deviation, so the microscopic misalignment of axons within the corpus callosum. The only problem is that when you look at this huge volume, then there are two contributors to the orientation dispersion of fibers in our measurement. One is indeed the microscopic orientation dispersion, similar to what Gary Zhang and then later on Hugo and Charlotte in their paper used to describe as the orientation dispersion of axons. But since our volume is so big, we also have a contribution from the macroscopic curvature of the corpus callosum within our volume. So how do we account at least for this confound, which is the macroscopic curvature of the corpus callosum within the volume? Well, to the aid comes the diffusion tensor imaging measurement, from which we can obtain the directions of the fibers within the corpus callosum using the so-called eigenvector, the main eigenvector of the diffusion tensor for each diffusion tensor imaging voxel. That gives us an idea of the distribution, of the angular distribution contribution of the macroscopic curvature of the corpus callosum within the volume. And what remains is to try to model out the microscopic misalignment of the orientation dispersion in the way that appears also in models like NODI. And in parallel, Matt actually obtained brain slices from the Brain Observatory in University of California in San Diego from Jacopo Annese, and estimated the orientation dispersion of fibers just looking from the post-mortem histology of those fibers within the corpus callosum. My model and his measurements actually gave us very similar numbers for the standard deviation or the kind of degree of misalignment of fibers within the corpus callosum. And I have to say that this is one of the studies about which I'm most proud, because not only we have here kind of support from histology, but I think that one of the things that is very dear to me is to try to combine measurements from different types of measurements. And here you can see a relatively successful combination of diffusion-weighted spectroscopy measurements together with diffusion tensor imaging measurements to give data that supports a model that gives results that are supported then by postmortem histology. By the way, this type of misalignment was also measured in the rodent brain, and in the corpus callosum, the misalignment also gives rise to a standard or angular standard deviation of about 14 degrees. So even in highly organized white matter tracts, one cannot neglect the orientation dispersion index, which is extremely important to us also when we go on and use models like NODI. I want to go to the clinical significance of this type of, let's say, compartment-specific measurements, and what can we say about the changes or modulation of diffusion coefficients of intracellular metabolites by disease processes. And this is a kind of a hypothetical slide. This is kind of what drives our hypothesis. And again, we said that there is a distribution of metabolites that is favoring, in some cases, neurons, for example, in the case of glutamate and acetyl-aspartate. But we said that, for example, myoinositol and choline are more driven to be found in glial cells. And we can kind of do an imaginary experiment whereby we introduce a pathological mechanism, like, for example, aggregation of tau, for example, in neurofibrillary tangles, or any sort of intracellular change that affect exclusively neurons. And we can ask ourselves, in what way is this change going to affect the intracellular diffusion of metabolites that reside overall in all cells in the brain? But since we are affecting specifically neurons, that we would expect that something will happen to the diffusion coefficient of neuronal metabolites. Well, one can assume that, for example, the increase in tortuosity because of the added neurofibrillary tangles might, for example, decrease the diffusion coefficient of those intracellular metabolites. Conversely, if we have a physiological / cytomorphological modulation of glia, for example, via glial activation, then this type of cytomorphological process may give rise to changes that are preferential or specific to more glial metabolites. In this case, in the case of glial activation as a result of inflammation, we may expect to see an increase in diffusion coefficients of those glial metabolites. And I want to briefly give you a couple of results on these type of studies. The first study that I was involved in clinical application of diffusion-weighted MRS was thanks to Danny Reich, and his then PhD student, Emily Wood. At that time, they were in Johns Hopkins University, and then Danny migrated together with Emily to the NIH, to NINDS. And we were interested in calculating the diffusion tensor properties. At that time, we were quite naive, so we were thinking, well, if we can choose a big box on a relatively coherent white matter tract, we might as well calculate the diffusion tensor of N-acetyl-aspartate on this particular chunk of the corpus callosum and see how the main eigenvalue or the diffusion of N-acetyl-aspartate along the callosal fibers behaves in healthy controls and what happens when we look at a patient with multiple sclerosis. And this is what is the so-called normal appearing white matter. And we saw a significant decrease in the intraaxonal diffusivity in those patients. And this is in stark contrast to the increase, as you can see, in the same type of measurement, which is the longitudinal diffusivity of water. So seemingly kind of a contradiction. We are measuring along the fibers, and here we see a decrease when we go to, when we measure the diffusivity of NAA in the corpus callosum, and here we can see an increase. Well, there is no contradiction. It is just a question, what do we actually measure? While we can be certain that when we measure N-acetyl-aspartate, we're measuring the intraaxonal space, when we measure the axial diffusivity of water, well, there is also a strong impact of the extraaxonal space. And it appears that what we see here is a winning of the demyelination effect, even though not very visible on the normal empiric white method, but still existing, compared to the intraaxonal disturbances that cause the decrease in this longitudinal diffusivity. This was, by the way, repeated in a longitudinal study. First of all, we replicated the cross-sectional studies, this time using the model that Matt, Buddy, and I came up. So this is a tensor, but here's the actual, what we call cytosolic diffusivity of NAA. And also here we have a decrease in MS and an increase in the water diffusivity. So decrease in NAA diffusivity, increase in water diffusivity. But we also detected the longitudinal effect, whereby after 6 months of we repeated the measurement, there was no change in the diffusivity of NAA in the corpus callosum for patients that were considered stable, but for patients that were considered active, there was a further decrease in this marker that we can now sort of with confidence say that it is a marker for some sort of intraaxonal damage in MS. This is not from my studies. This is from a study from a great collaborator, Francesca Branzoli, that you see here on top, together with a student, Guglielmo Genovese. Francesca was my postdoc in Leiden and started being an independent researcher in the Institute for Spinal Cord and Brain Research in the Pitié-Salpêtrière Hospital in Paris. And Francesca looked also at the corpus callosum. Well, corpus callosum, we MR physicists love the corpus callosum for various reasons. Here in this case, it is the corpus callosum of mice that are supplied in their food with cuprizone. And cuprizone is an chemical that induces demyelination or reversible demyelination, and thus it is used in many preclinical studies of demyelinating disease, such as multiple sclerosis. And I want to show you those magnificent results that Francesca and Guglielmo came up with respect to changes in the diffusion coefficients of intracellular metabolites in the cuprizone model. And you can see that when you look at the decay curve of N-acetyl aspartate, the neuronal component, there is absolutely no change in its decay upon diffusion between the cuprizone model and healthy controls. However, looking at glial components like myoinositol and choline, there is a very strong differentiation between what happens with the cuprizone model, which is an increase in the diffusion coefficient. There is a kind of a tiny thing that you can obtain with the creatine, but there is nothing statistically significant there. So there is a sense that in this case, the demyelination and the kind of reversible demyelination cuprizone model, the effect that you see here is not an axonal pathology, or we do not detect any axonal pathology, but we do see the immune reaction to what is happening during demyelination, resulting in an increase in the diffusion coefficient of glial components. This is just a snapshot into their study. There is also immunohistochemistry validation of it, so I recommend everyone to take a look at this very, very interesting study. Meanwhile, back at the ranch, Ece Ercan, who was my PhD student at that time, and myself were engaged in measuring similar measurements that we did with EMS, only with another inflammatory disease, which is systemic lupus erythematosus, and in particular, SLE patients with neuropsychiatric complaints. Our hospital is the National Referral Center for such patients, and thus we have quite good access to research on lupus with neuropsychiatric complaints. And again, in this case, we stuck to the corpus callosum, not because of any particular hypothesis, but just because we were kind of used to do it. And in this case, we again did not detect any change in the diffusivity of N-acetyl aspartate between NP-SLE subject, SLE without NP, and healthy controls. But in the choline, which again, we saw also in the neuroinflammatory model in the cuprizole model, we did detect a significant increase in diffusivity in the SLE subjects, but also an even more significant increase in the diffusion of creatine. And this is something that is interesting because it will link perhaps, again, we don't have proof for that, but will link perhaps to the metabolic meaning of the diffusion of creatine that we will see later on. I will conclude this part by mentioning the latest, la dernière cri of those type of measurements. And this is one of the reasons why I am actually in Brighton currently, but will be in Brighton as academic director of CISC, is this beautiful collaboration that started with Mara and Neil and Iris, and together with Francesca, to look at the changes in diffusion coefficient of metabolites in the brains of people that were infused with lipopolysaccharides, creating a model for systemic inflammation that we hoped would eventually cascade into neuroinflammation. And from those two measurements that you can see here, we have a measurement that was performed in a big voxel in the thalamus, and here in white matter. When you look at white matter, there is no difference in the diffusion coefficients of choline or creatine or NAA between LPS and controls. Well, control, these are the same people that are the controls for themselves, so control and LPS conditions. On the other hand, in the thalamus, where you can see that there is a significant increase in the diffusion coefficient of choline, and nothing at all with the N-acetyl aspartate. And this gives us the impression that what we see here is selective activation of microglia in the thalamus. This is, of course, a kind of an initial study. You can see very few subjects, but even with very few subjects, the statistical significance is surprisingly high. And Neil and Mara and others continue to look also at correlations with behavioral outcomes. And even here, there is, for example, an interesting correlation between the apparent diffusion of choline and scores related to negative mood and total mood score. So interesting also on the link between measurement and actual outcome. I want to switch to the, I hope I still have time. Chris, you keep me in check? I hope so.
I'll flag you at 10 minutes.
Okay. So in this case, I will try to kind of go relatively quickly through the functional studies. I did prepare a couple of slides related to my past as a seeker for physiological specificity in functional MR. This is a successful example of such research. It's my PhD thesis about kind of an alternative or proof of concept of an alternative method for detecting metabolic generation of metabolically labeled water. So essentially the MR alternative to oxygen 15 PET, if you want. This was actually what brought me to do my postdoctoral research in University of Minnesota. So this is a good example of such research program. A bad example is what I insisted on doing for about 2 years, which is to try to detect neuronal activity in the rat brain with sodium MR. These were some of the toughest experiments that I performed as a postdoc, totally against the advice of my postdoc advisor at that time, which is Sangi Kim. By the way, this is my PhD advisor, Gil Navon. This is Camille Ugorbil, who is the director of the Center of Magnetic Resonance in Minnesota. Those measurements that tried to distinguish or to separate the different sodium pools in tissue by injecting the rat with what we call a shift reagent, so a reagent that shifts the resonance frequency of sodium when in contact with sodium in solution. You can see the situation before the infusion. We just have 1 peak of sodium. And you can see that after infusion, we have something that I then interpreted as the vascular pool and the intracellular pool, extracellular pool. And I injected the poor fellows with bicuculin to create a kind of a global activation and try to figure out why is it that those peaks change in frequency, and hypothesized that the increase in exchange rate between the extra and intracellular sodium creates this kind of rapprochement between the extracellular and intracellular pool. But I think that those results are not indicative of actually what happened. And this is just to give you an idea that someone can have good ideas, but bad experimental design, and eventually this type of approach, unfortunately, I had to abandon it for a variety of reasons, but not before I killed many rats, and this is why I selected to add it here, that the road to rat hell is paved with very good scientific intentions, but one has to be very careful about hypotheses and how to check for them. But there are, legitimate approaches to try to get closer to neuronal activation. The age-old C13 NMR and phosphorus NMR measurements that measure metabolic turnover of glycogen and oxidative metabolism, glucose and oxidative metabolism. One can also recur to standard proton MRS, using it as a functional tool to look at changes in steady state concentrations of excitatory and inhibitory metabolites, but also in metabolic products like lactate. And one can even go the arduous way to try to do direct mapping of neuronal currents using the phase of the MR signal. But one of the suggestions that was done by one of the founding fathers of diffusion measurement, which is Denis Lebihan, that you see here, is that when you do functional MRI experiments, but instead of using the standard bold fMRI paradigm, you use diffusion-weighted MRI, the response, the tissue response to the stimulus happens at an earlier stage than the bold signal. This type of experiments is experiments that few people did. Denis started with them. There was a huge controversy on actually what is that we are measuring with this type of measurements. And regardless of whether what we measure here is indeed a structural cellular reaction to a neuronal activity, curiosity is definitely activated by it. And this is something that made me, for example, very curious. And I said, okay, if there is a diffusion effect, why don't we try to measure it with diffusion-weighted MRS and come up with a paradigm in which we measure diffusion of metabolites in the course of visual stimulation experiments, a kind of a block design experiment, and try to understand what is that we're seeing. And what we saw, surprisingly, is that there is a significant increase in diffusion coefficient of both N-acetyl aspartate, even in choline, even though this blob is kind of strange in terms of the fact that it kind of starts way before the stimulation and kind of dies way after it. But when you look at the time course of the changes in the creatine diffusion coefficient, it seems to be very well correlated with the stimulus. And this is both reflected by the p-value of the change, the actual change in ADC between the two conditions, but also in the correlation with the boxcar kind of stimulus, and just kind of measuring the cross correlation between response and stimulus. And it is not clear what we're measuring. Are we measuring changes in a metabolic effect on the diffusion of creatine, which may be, for example, a result of imbalance between creatine and phosphocreatine that have significantly different diffusion coefficients, or it may be still a structural difference. So we don't have a clear evidence to that. And for that is, I try to go further with a, let's say, more specific experiment, which was introduced by Daniel Topgaard from Lund University, depicted right here. And this is a so-called filter exchange spectroscopy experiment. Filter exchange experiment in general can be done in imaging mode and can be done also in spectroscopic mode. And the principle is quite simple. If you have two compartments, one is relatively free diffusing and one is restricted, then if you apply a strong diffusion weighting module, you kind of eliminate the contribution of the free diffusing compartment. And then if you let the system to evolve in time by introducing what we call the mixing time, if there is exchange between the two compartments, then the fast diffusing compartment will be repopulated by spins that cross the membrane. And then when you detect the situation with your diffusion weighted module right at the end of the sequence, you take a snapshot of what is happening here, which will be dependent on the exchange rate between the two compartments and also on the mixing time. So this is an interesting experiment, very difficult experiment. We can then compare everything to the measurement without filter, so essentially just measuring the structural component of the system. And quickly, I kind of programmed the sequence on our 7 Tesla scanner, recruited a master's degree student, Casper Wolf, who you see here, and we performed measurements on very few subjects, essentially very complicated measurements with 4 different conditions, and for which the results are totally non-conclusive. We have very few subjects, and we have very few significances in this type of results. But one thing that I do want to kind of insist on is that we do have, first of all, so it's a complicated graph, but this green arrow here tells you what happened to the diffusion coefficient of creatine between the resting condition and the stimulated condition. You can see a rather steep increase in diffusion coefficient that is quite specific to the creatine and not seen for the NAA. And I suspect that what we see here is indeed an increase in some sort of an exchange between creatine and phosphocreatine, or between 2 different compartments where both of them reside, for example, mitochondria and the cytosol, that is accelerated in the case of stimulation. This is one of the studies, for example, that I would love to continue to do when I am firmly positioned back in Brighton. I will very quickly go through my plans for research in CISC. Chris, can I have the next 10 minutes? Is that okay?
Yes, absolutely. Yes, of course. Okay,
thank you. Because I think it is important, and I'm sorry that I kind of spent too much time talking about the past and not so much about the future. But first of all, kind of briefly to say, how do I envision my role as an academic director of CISC? So I think that the long-term goal is simply to try to forge a kind of a USP, unique selling point, or unique identity for CISC that is based on a combination of the methods or the methodological expertise within CISC and the science that is going on around campus, and either already uses CISC or potentially can use CISC. And again, I say it with all the humility that is necessary to say this, because here I come after 11 years of active CISC with Mara at the helm. So a lot of the footwork has been already done. So I am coming in a situation that's quite different from the situation where I came here in Leiden, or when I came to Boston University, where there was no imaging center. So I have to thank Mara and Hugo and all the rest for the fact that I'm coming already to an existing situation, and all I need to do is to create a delta. And the way that I'm going to try to introduce this delta is, of course, to develop and validate and implement tools and methods that match with the interest of researchers on campus, and to try to expand the user base of CISC via, of course, internal collaborations, but also external collaborations, both in the UK and also, of course, abroad. And lastly, but not least, is to increase the role of CISC in education. And I think this is extremely important also in attracting talent from the younger generation to be involved in imaging. All of this, of course, trying to be sustainable with respect to the available resources that we have, both in personnel and equipment. I'll immediately jump into the two kind of, let's say, main lines of research that I would like to establish as soon as humanly possible when I am in CISC. One is based on strong collaboration with the group in which I reside right now, which is the Leiden University Medical Center. You see here Andrew Webb, who is the center director, Tom O'Reilly, who is his PhD student. And I should say almost single-handedly, both of them started the program in low-field MR. What is low-field MR? Well, you all know 3 Tesla and 1.5 Tesla. Here we're talking about 55 millitesla. This is the magnet. This is the console of the scanner. You can see here, well, I should say an internal volunteer scanning his knee. You can see some knee images here on the console. And here you can see already some initial brain images from this home-built magnet, both T1-weighted images at various inversion times and T2-weighted images giving rise to T1 maps and T2 maps at 55 millitesla. And low field is a kind of a strange thing, because when you think about it, MRI started with low field. Now we're back at low field. But the thing is that with the maturation of methodology in MR across the whole pipeline, so from hardware to image analysis, we can now start thinking about what can we do at low field that gains from the fact that we do the measurements in low field. So first of all, we can think about portable scanners that can be brought to remote and underserved locations. We can think about multiplying scanners and putting them in the GP office in order to have screening for diseases that will not require you from going to a hospital in order to get scans. And of course, to overcome counterindications like implants that make scanning on high field scanners exclusive for people that can be scanned at those scanners. So this gives us opportunities to develop new hardware and system design, new image acquisition methods, new image analysis methods, and also use it as an educational design. This is Chloe, who is now going to be my remote postdoc, if you want, in the low field scanner here in LUMC, and will be hopefully also the bridge with a similar program that we will start in Brighton. And the work package that we are funded to do so, this is all part also of funding that we received from the Dutch government to perform. And my role in this is to try to develop spectroscopic methods for low field MR. And again, I don't have much time, so I'll really gloss over it. But one of the targets for spectroscopy at low field is lactate, because lactate is a marker for several neurological diseases. And I set my eyes on hypoxic ischemic injuries in newborn babies, which are particularly fit for low field MR in the sense that they fit physically also within the scanner. I just want to tell you about our initial methods. We just showed already in a very recent publication that we can obtain a type of spectra that is independent of field and can give us evidence of lactate, in this case in ping pong balls in a small coil, but hopefully also in vivo situations in real measurements. The other line of research that I think would match fantastically with what people on campus are doing is to use what has been established in Yale as Deuterium metabolic imaging as a potential alternative for FDG PET. And again, very quickly to browse through the slides, the group in Yale is led by Robin de Graaf, a Dutchman that is many years abroad, a luminary in everything magnetic resonance spectroscopy. But here again, a blast from the past. Deuterium was used many, many years in the past for spectroscopic measurements, and now it's making its comeback as an alternative for measurement of in vivo metabolism. One can ask, what are the advantages of measuring it with deuterium? Well, there are a couple of interesting advantages. What are the alternatives? The alternatives are to use PET. Well, we know that this is indeed a possibility, but we're looking for a method that can be integrated in MRI, maybe will be cheaper than PET, and also not be as invasive as PET. The use of deuterium can be done in steady state. And in this case, you can see, for example, things that we cannot detect with proton magnetic resonance spectroscopy, like glucose. So we can get the distribution of glucose in the case of a brain tumor. You can see here that there is a decrease in glucose and a decrease in glutamine and glutamate, but increase in lactate, kind of exemplifying the so-called Warburg effect in brain tumors. Here is a kind of a spectroscopic evidence for that. But so important to say that deuterium has been approved for studies in humans. One can infuse with deuterium labeled glucose, or even start with infusion or drinking orally deuterated water, or what we call heavy water in this case. This can be implemented with standard spectroscopic imaging techniques, required, of course, a radiofrequency coil that costs quite some money, but not an exorbitant amount of money. And again, the advantage over proton spectroscopy is that it allows studying fluxes or metabolic fluxes rather than steady state situation that is the only thing that we can do with standard MRS. We can detect lactate, but we cannot detect the metabolic rate in which the lactate concentration is changing in time. So this is one methodology that I think fits very well with many of the people on campus that are interested in studying metabolism. And I want to go very quickly through a couple of, let's say, fast ways to adopt methodologies that exist already on Siemens platforms with groups that I already have strong collaborations with, and can be, let's say, in the course of a couple of months already implemented in CISC. One of them is the method to target what we call microscopic anisotropy in tissue using so-called B-tensor encoding. This is the brainchild of the group in Lund. Philip Szczepankiewicz and Markus Nilsson and Carl Frederick Weston, who is in Brigham and Women Hospital in Boston, are the founding fathers of this methodology. Here you can see, for example, the histology of two different types of tumor. This is the case of meningioma, where cells are essentially kind of, you can envision them as kind of mini fibers that are kind of randomly oriented within the measurement volume. In the case of glioblastoma, the microscopic feature is more, let's say, spherical in feature. And you can see that the difference in microscopic anisotropy in the tumor is highly different in the case of meningioma and glioblastoma. Gives us hope to use this type of microscopic measurements also in clinical applications. Philip already agreed to come over to CISC anytime we want him to come over to implement the methodology on the Prisma scanner, make it available to researchers on campus. And this is Markus right here. The other method that I'm extremely excited about is so-called magnetic resonance spectroscopy fingerprinting, which is essentially a way to obtain not just spectroscopic information about concentration of metabolites, but also about relaxation properties of metabolites that are also modulated by intracellular phenomena that can be modulated in disease. The founding father of this type of measurement is Asaf Tal, who is in the Weizmann Institute in Rehovot in Israel, again, a close friend and collaborator, and he already gave his okay to bring us this method to CISC. And the last such method is a method that James Stone actually expressed interest in and hinted at that when we had our conversation when I came for the chat with the CISC people back in May this year. And this is the indirect detection of glutamate via its interaction with water protons gives rise to the possibility of mapping glutamate concentration in vivo, not using spectroscopy, but using imaging. And this is from the paper of Ravinder Reddy, who is again the founding father of this method in UPenn, and Ravinder also, Ravi, his friends call him, gave his consent to give us the methodology to be implemented on the scanner. So I rushed very, very quickly through my plans, and I hope that something of that remained with you guys. I want to also pay tribute to the place that I'm still employed by, soon to be to say goodbye to it. This is Leiden University Medical Center. This is the not so beautiful building of the hospital. This is the very beautiful building of the so-called academic building, the Akademiegebouw, the building of the first university in the Netherlands, given as a gift to Leiden for their courage in their revolt against the Spaniards by the founder of the Dutch Republic, William of Orange. These are researchers associated with Leiden University, many of them related to MR. This is Hotel, after which our center is called. This is Heike Kamerling-Onnes, who is kind of single-handedly responsible for the fact that we can do measurements of ultra-high field, because he is the one that discovered superconductivity. And the last but not least is one of the most important natives of Leiden. This is Rembrandt van Rijn, and this is his famous anatomy lesson of Dr. Tulp, which he taught us all that we can do invasive anatomy using postmortem studies, maybe giving us some sort of an inspiration to do them not so invasively using MRI. By this, I thank you very much for your attention and apologize for the lengthy talk.
Thank you very much, Itamar. Fascinating talk and a huge amount of information. I'm aware that people might want to dip off, but if people want to stick around and ask questions to Itamar, by all means, I can keep the Zoom open for a bit. If Itamar has time to stay.
I definitely have time, and I will just stop sharing so I can see also people.
So does anybody have any questions? Perhaps not.
Guys, please, I mean, there will be, of course, opportunities on campus, but I am here for you guys, so just ask whatever is on your mind, from science to non-science to everything.
I'll briefly ask, do you know if, you know, the microfield MRI scanner, is that possible? Is it possible to do functional scanning with that, or is it just structural?
I'm sorry, I'm kind of losing connection.
Oh, I was just asking, with the microfield MRI scanner that you talked about, is it possible to do functional scanning with that, or is it strictly structural?
No, actually, there is a very interesting, there are two different approaches for functional studies. One was introduced by an Englishman, Ben Inglis, who is in Berkeley University. And it's a very ingenious way to do functional studies at low field. Again, you will not have the advantages of So it is obviously not based on P2 star, because everything that is related to differentiating species based on disturbances of the magnetic field shrinks down. So this is not P2 star related, but based on alternative mechanisms. He actually went even lower in field and showed that in several microtesla, you can actually do functional studies. So there is hope also in very, very low field. There is also a recent study by the group at MGH, I don't remember what was the guiding principle there, but there is hope. Again, you have to ask different types of questions and expect spatial resolution to be compromised, but you can ask questions, functional, and obtain functional signal, definitely.
Very cool. Iva has a question.
Hi, Itamar. Thanks for the great talk. It was great to see all the various things you've done over the years. But I had a question about the MR spectroscopy and diffusion measurements. So I was just wondering how much kind of spatial specificity you can get with the sequences you're using, because some of the boxes were quite large. Now, there are particular kind of modeling challenges that arise as a consequence of partial voluming.
You're absolutely right, of course. And the major limitation is spatial resolution. And there is no question about it. So you have to somehow infer, or so one way to overcome it, and this is something that we did in our latest endeavor, microstructural endeavor, is to say, okay, let's, for example, talk about partial voluming between gray and white matter. What you can try to do is to try to get correlation. So you can measure with your big volume in a volume where the percent of gray and white matter would change dramatically. And you will have enough points on the graph that will allow you to infer this type of microstructural inferences based on the amount of gray and white matter that you have in the volume. So that we do, of course. So for each such measurement, we also acquire anatomical image. We correlate it with the volume. We segment it within the volume. We get information about the percent tissue, and we come up with some sort of correlations. That doesn't solve everything, but at least it mitigates a little bit the question of partial volume. Yeah, you're absolutely right. We cannot win them all. So, that's one of the things that I was in Korea. That was, I think, more than 10 years ago. There was at that time there was an initiative to have a 14 Tesla scanner in Korea. Professor Zhou, who was at that time in a small university called Gachon University, wanted to lead this kind of effort, and they brought all the luminaries. Seiji Ogawa was there, and then Levian, and people from the industry to say what do they envision about ultra high field 14 Tesla. And one of the things that Seiji Ogawa actually said is, maybe at very, very high field, we can treat signal from low concentration species like sodium, like metabolites, like what we treat now, the water signal. So again, maybe at 14 Tesla, we'll be able to get to the resolution where we have now at, kind of mid-field, even though I suspect not. The differences in concentration are so big that we will always be relegated to voxel sizes, the centimeter scale. But at least we can try to increase the specificity. That's definitely true. Another possibility is, of course, to try to somehow use other physical properties like differences in relaxation, etc., to try to maybe filter one type of tissue from the other. Fortunately, with metabolites, it's a little bit difficult to do because you anyway don't have much signal to try to get as much population as you can within the volume.
Dora, do you have a question?
Just a practical question, actually, because I was very impressed by the different methodologies and the possibility of all those. So I was just wondering, knowing that in CISC is only 3 Tesla, of course, there are ways of collaboration, but what do you think are the sort of methods that you would like to start, knowing what the possibilities are at CISC?
It's a very good question. Yeah, it's an excellent question. And I think, 3 Tesla is a good field. Is a good field in a sense that it's higher than 1.5. That's one good thing about it. The flip side is that it's not high. It doesn't get into the troubles of ultra high field that requires, let's say, spatialized methodology. So let's say from out of the box type of scanners, 3 Tesla is a relatively ideal scanner. Now, the nice thing about the 3T at CISC is that everything else except for the field is, as I should say, state of the art as possible, in particular, the gradient system. And the gradient system essentially tells us that everything that has to do with diffusion can be done essentially in a way that is, well, I shouldn't say completely comparable, but on the same ballpark level as scanners with higher field that have, let's say, lesser gradient system. So yes, you're absolutely right. And I think we'll have to somehow kind of test and take some risks also, because I kind of glossed over it, but for example, the Deuterium study that I mentioned that I want to bring, the study in Yale was done at 4 Tesla. So 4 Tesla is not the difference from 3 Tesla. And the concluding sentence that they put in their paper is that the proliferation of 3 Tesla scanners actually is a good indicator in terms of the possibility of implementing this type of measurements in a clinical setting. So they think, well, they also think a little bit like used car salesmen. Of course, one has to remember that because it is their method and they try to sell it. But I think that knowing the system at Yale, which is indeed a 4 Tesla, so a little bit higher field, but it's a very old scanner. So I think that in terms of the RF chain, for example, the Prisma in CISC is better. In terms of the gradients, which has quite a bit of impact on the spatial encoding efficiency of the deuterium signal, we are better than Yale, well, 4 Tesla in Yale. So there are several, let's say, hardware entities on the balance, which I think allows us to be quite optimistic with respect to implementing, for example, the metabolic imaging, the deuterium metabolic imaging on 3T.
Okay.
Preferably on 70, but if you can't, then do it at 3T. You pay a little bit with resolution. That's definitely going to happen. But, not always spatial resolution is that important. Sometimes, you can get by with, let's say, kind of more blobbish measurements. And, I don't want to sound, too optimistic about machine learning and things like this, but there are things that, and that's also true also for the low field MR, is that one of the hopes is that whatever you cannot obtain with the acquisition, you can actually obtain with the image analysis and signal processing. And maybe analysis methods that will be adopted from compressed sensing and from machine learning and other kind of methods that operate in a situation where there is scant data to work with, will work well with this type of methods. But future will tell.
Yes, thank you.
Thank you.
Great, thank you very much. Yeah.
Balazs and colleagues, do you have a
yes? Prince has a
Hi.
question. Yeah. Hello. We're actually watching from CISC. Itamar, I'm sorry, I haven't met you yet. I'm one of the clinical research fellows at CISC.
I know about you. I haven't met you yet, but it's a pleasure to meet you also in such a remote situation.
I mean, I was really very busy and I haven't really listened to all the presentations, but I'm sure you are aware that I'm trying to do some work with DW-MRS on the hippocampus. And we're also looking for changes in oxidative metabolism and things like. I was just wondering if this was going to be possible, using DW-MRS on the hippocampus. I mean, I've been placing the voxels. It hasn't been a perfect fit on the hippocampus, but I'm more interested in the head and the body. But I just wanted to get your pick on it.
Am I allowed to be honest?
Yes, very frank.
Okay, I'll try to be honest. Well, let me take your question one step back and ask, is it possible to do MRS on the hippocampus? Well, the answer is yes and no, in the sense, of course, you can put your voxel on the hippocampus and try to get your spectroscopic signal from it. History and experience tells us that MRS on the hippocampus is difficult. It's difficult because it is difficult to get good shimming on the hippocampus. I'm now being privy to a lot of hippocampal MRS data through a collaboration with a good friend in Lund. So Oskar Hansson, who is a neurologist in Lund, has a huge data set of MRS in Alzheimer patients in the posterior cingulate and on the hippocampus. And the difference in quality of the spectral quality between the posterior cingulate and the hippocampus is just huge. I would say that 50% of the spectra from the hippocampus had to be thrown away just because of low quality. And that makes it really difficult. Again, one can be optimistic and try, and if you have enough patients and you have enough data sets to throw away and to rely on 50% of them, then you might be successful. But I would say that one has to be careful. And of course, I will be extremely, extremely glad to help. But based on what I see, try, get it. And if you see that you cannot get good shimming, for example, and that the quality of the spectra is insufficient, then it is insufficient. So have you acquired already just standard MRS spectra on the hippocampus?
I think, yes, somewhat to an extent, but I think the numbers we have are really low. So I have 9 MS patients.
Well, just share with me and I can just take a look, do some quality inspection, see if it's good enough to continue on to think about adding diffusion weight.
Okay. Thank you very much. Thank you.
My pleasure.
Okay, so I don't see anybody else with their hands up, so I think maybe we should end it there. So thank you very much, Itamar, and thank you everybody else for coming along, and welcome to Sussex.
Thank you so very much. I'm looking forward to seeing each and every one of you personally when I'm there, and that will happen very, very soon. So thank you so much for being present here. Thanks so much. And thanks, Chris, for organizing.
You're welcome. Thank you.
Bye.